Hello everyone, welcome to MetaCell's webinar on building multiscale brain models. I'm Matteo Cantarelli, I'm the CTO of the company, and let me first of all thank you for taking the time for joining us. We are pleased to have Dr. Salvador Dura-Bernal from the State University of New York with us today. Salvador is a research assistant professor at SUNY Downstate and principal investigator in 3 research grants. He has published 24 peer-reviewed papers covering computational neuroscience, bioinformatics, robotics, and machine learning. His current research focuses on the development, parallel simulation, and analysis of large-scale biophysically detailed circuit models, and for this purpose, he has developed NetPyNE, and we'll talk about it. Using NetPyNE, Salvador has built a large-scale detailed model of motor cortex microcircuits, which can be used to study neuronal coding mechanism and treatments of brain disorders. So, Salvador, thank you for joining us. How are you?
I'm good. Thanks, Matteo, for the invitation.
Excellent. So, we'll have first just a few slides about MetaCell. MetaCell is a software company that builds software for neuroscience. We turned 7 just this last July. A lot of the software that we build has to do with visualizing and sharing data and models online to help researchers and teams to collaborate together. Throughout the webinar, we're going to have some interactive polls where we'll be asking you some questions to get a better sense of what interests people that are listening today. And so there will be a pop-up window that comes out from the webinar software, and you will be able to select a few options to answer these questions. So as MetaCell, we work with a large variety of organizations spanning from universities to the industries, pharma companies, small companies, and non-profits. So the first poll I would like to launch is just to understand a little bit the breakdown of the room and to understand if you work on academia or in the industry. So let me try this. So you should have a window that comes up, and I see real-time votes appearing. Almost everyone has voted. Another few seconds. All right, here we go. So you should be able to see the results here, where 75% of the audience today comes from academia, 19% from the industry, and 6% others. Thank you for voting. This is helpful also so that we can adjust the target of the presentation. So what are we going to talk about today? Today we're going to talk about multiscale brain modeling using NetPyNE. NetPyNE provides a programmatic and graphical interfaces that facilitates the definition, parallel simulation, and analysis of data-driven multiscale models. NetPyNE is developed at the State University of New York in the NeuroSim lab and is funded by the NIH and the New York State Spinal Cord Injury Board. MetaCell worked together with Salvador and the NeuroSim lab to build a user interface for NetPyNE. We will have a demo later on. And before we get into the details, Salvador, of NetPyNE, user interfaces, and whatnot, why don't we take a step back for the audience and talk about what it means to create a computational model and why do we want to do this. So I'm going to give you control of the presentation so you can share your slides. And fantastic.
So why do we do these computational simulations of the brain? So the basic idea is that for many years now, we've been collecting a lot of experimental data from the brain, from different scales of the brain, both at the single cell level, the morphology of the cell, biophysical properties of the cell, how they connect together in small circuits, and how they connect together along the entire brain. And the idea of simulation is to help understand and make sense of all this experimental data. So to enable this, what we do is combine all these experimental data into a single sort of theoretical framework, a simulation, that then we can use to explore the behavior of the circuit, do different tests on the circuit, for example, try different experimental treatments, and try to better understand how the brain works. So the simulation allows us to have a very controlled environment and perform experiments that are very difficult to do in the lab.
Okay. And do you want to, I think this was a great introduction. Do you want to tell us then about the motivation behind NetPyNE, how it can use to build models, and how it can help serving these aims?
Sure. So basically, I started developing NetPyNE because I was starting to do a modeling project with Northwestern University, and we had to build a very complex model of motor cortex microcircuits. One of the issues with the brain is that it has multiple scales that interact together. So here we have a diagram showing we have the sort of large scale dynamics of the brain, where different regions of the brain interact with each other. If you zoom into one of these regions, you actually see a lot of complexity. For example, cortex has 6 different layers with different cell types in each layer, and they all connect to each other in very complex patterns. And if you zoom into one of these specific neurons, the basic units of the brain, you will also see a very complex machinery, all these molecular interactions that basically dictate the behavior of the single cells. What is even more complex about the brain is that all these 3 scales interact with each other. So the molecular interactions that happen inside the cell affect the behavior, which affect the network activity and affect the whole brain activity, and ultimately the behavior of the human being. So in this project that we had to do, building a very detailed model of motor cortex, we had experimental data from all these different scales. So we had, for example, the morphology and biophysics of different cell types. We had how they connected to each other at the local level in the microcircuits, and as well in different parts of the brain, so inputs from different regions of the brain and how they project it onto the cells. And we also had data about the biophysics of these cells. So in order to make sense of all this data, I started building a sort of software that would help me do these simulations. And this software slowly converged into this NetPyNE tool that we've made now publicly available. The other motivation was making this modeling easier than it was before. So before, you basically have code, computational code, Python, for example, which is mixed up with all the parameters of the model. So if you look at any of the existing models, for example, for NEURON in ModelDB, you will see lots of different implementations in, for example, Hawk or Python. And it is usually quite hard to unravel the parameters, the biological parameters of your model from that programming code. So we wanted to do it in a way in which the parameters of the biological parameters are separate from the actual implementation, from the actual programming. This also enables to add sort of a standardized implementation, so the user doesn't have to worry about how to implement all this code. They just specify the parameters of the model, the biological details of what they want to build, and in the back end of the software, there is a standardized implementation which has already been tested and is ensured to be efficient, that implements everything that is needed. So this makes models much easier to read, to interpret them later on, to modify them, and to share them with other people. The third motivation for us was making it easy to parallelize models. As we increase the size and complexity of these models, it becomes necessary to parallelize them, so to run them on, for example, supercomputers across many hundreds of processors. But this process of parallelization is not trivial, especially in the neuron simulator. You have to add a lot of specific commands, and you have to know how to deal with the parallelization design. So, for example, you have to distribute all the computations across different nodes, for example, different cells in different processors. You then have to gather back all this information in order to analyze and visualize the output of the simulations. And you also have to ensure that the results of your simulation are independent of the number of processors that you use. You have to ensure that the results are replicable independently of how many cores you're running on. So all this complexity we wanted to hide from the user, and that was also another motivation for developing this tool. So I'm going to show here a sort of structured diagram of how NetPyNE looks, the different modules that compose NetPyNE, and then we will look a bit more into a couple of them. So the first big module is what we call high-level specifications, which is basically where you define what you want to see. So NetPyNE, as we will describe later, it's a declarative language. So instead of specifying how to do everything, you just say what you want to do. So here we can define all the parameters of your network. You can import existing cell models, for example, and you can also define parameters of the simulation. So the duration, how long do you want to simulate your model for, etc. The next step is what we call network instantiation. So from these high-level specifications, which could be, for example, a probability of connection between two populations, you go into the second step, which is actually generating the network. So from those specifications, you generate a specific network with hundreds or thousands of cells and up to millions of connections. Once you have that network, which is instantiated in NEURON, the neuron simulator, we go on to the parallel simulation step. So again, NetPyNE takes care of all this process, distributes all the cells and connections across different nodes, and then runs the simulation using the neuron simulator and gathers back all the information that you need to process your results. The final step would therefore be the analysis and saving step, where you have lots of built-in analysis plots that you can use to visualize your data and explore your results. And you can also save to common formats like MATLAB or Python formats, HDF5, so that you can analyze those results in other tools. Importantly, we also have this ability to export to UML that we will talk about later. And finally, we have a module here at the top that we call the batch simulation module, which allows you to run batch simulations, so groups of simulations that, for example, explore the parameters in your model automatically. So they submit jobs to supercomputers, and they allow you to explore in more detail your simulation.
Well, I'm sure everybody is like amazed by how many things NetPyNE can do at the different steps, the instantiation, the parallel simulation, and also the analysis. So can you maybe say a little bit more, Salvador, about, so we have there on the left-hand side of the diagram, neuron cell models that are being fed into the high-level specification. So how does the high-level specification compare to the traditional way that you can define models using NEURON? I think that would be important to understand.
Sure. So let me just mention briefly what is NEURON before, in case people don't know about it. So the NEURON simulator is a widely used simulator. It covers all these scales that I was talking about, from the molecule to the cell to the network. And it has been used in over 400 or 500 models that are available online. There are over 1,500 papers that use NEURON. And it is also being used by large simulation projects like the Human Brain Project or the Allen Brain Institute to build very large-scale detailed models of the brain. However, NEURON is still relatively complex for the average user, and you have a sort of steep learning curve until you can build especially these very complex brain circuit models. So here's an example of how you would specify creating a connection in NEURON. These 6 or 7 lines are required to create a single synapse. what we did is in NetPyNE is sort of abstract this. So we made what we call a declarative language, which means that you, again, don't specify all of the instructions to be executed, but you say in a sort of more human readable way what you want to create. For example, here we want to create a set of connections from one population, say the sensory population, to the motor population, with a probability of connection of 50%, a weight, a strength of the connection, a delay of connection, and using a specific synaptic mechanism, for example, AMPA or an MDA. What NetPyNE does in the backend is create all these connections, which could be potentially up to thousands of connections with this single instruction here. And again, it's a sort of standardized implementation in the backend, so you don't have to worry about errors, and you don't have to worry about implementing it correctly for parallelization or any other issues that might arise by not knowing the specifics of the language that you're using. The third level that we have added is this graphical user interface. So we have converted this sort of high-level way of defining a network model into a graphical interface, which makes it even easier for people who don't know, for example, what parameters are available, to create network models easily and rapidly. So let me say a bit more about what things can you define with these high-level specifications. And it basically covers all the different aspects of a detailed network model. So you have from populations of cells, where you can define the cell type of the cell, the number of neurons or the density of that population, the net density of neurons, and the spatial extent. So you could, for example, place different populations in different regions of a network or of some tissue that you're simulating. You then, of course, have all the properties of the different cell types. So you have the morphology, you can have very detailed morphologies, you have all the biophysics, and you can even define molecular level processes that we're going to see in a minute. You can, of course, define all the parameters of synapses, and you can add some stimulation to the network, so artificial stimulations, for example, spike generators simulating inputs from other parts of the brain. You can have current clamps, and you can specify very complex spatiotemporal patterns with a very easy interface. One major feature that we have is these connectivity rules, where you define the connectivity of the network, which is usually very complex because it's obtained from experimental data and from biology. And you have very easy rules to define all this connectivity. And of course, the configuration of your simulation. How long do you want to simulate for? What do you want to say? What do you want to plot? Etc. So I'm going to focus on just two of these features that are basically at the opposite ends of the scales. I'm going to say a bit about this molecular reaction diffusion support that we have. We're just finishing adding support for this, and it will be released in the next version. And this is a feature from NEURON, which is now integrated into NetPyNE. So you can declare properties of this molecular level together with all the other features of the network in a sort of integrated framework. So this molecular reaction diffusion tool allows you to create, for example, define the diffusion of ions both inside the cell and outside the cell. For example, ions and proteins like calcium, potassium, IP3. And you can define their diffusion and reactions within different structures in the cell. So neurons are not simple structures, as I said before. There are a lot of different organelles inside of them, and they result in very complex reactions that affect the behavior of the neuron. So for example, you could define a region which corresponds to the endoplasmic reticulum or mitochondria, and then you can define many processes happening along this cell. So for example, things like phosphorylation, buffering, second messenger cascades. So here in these figures, you see the diffusion of calcium here in the left. You also see extracellular diffusion of potassium, the blue figure in the middle. You see the different organelles in the cell. And on the right, you see a second messenger cascade, which is also being modeled in NetPyNE. So this is important because, of course, all these processes at the molecular level are going to affect the firing of the cell and ultimately the behavior of the network. The other major feature for NetPyNE was connectivity. So we spent a lot of effort trying to make this as flexible and encompassing as possible. So we have the option to define connectivity, for example, as a function of probability, convergence to a cell, to cells, divergence from cells, which are common features that you find in experimental papers, or you can define custom connectivity rules. You can also define parameters such as this probability or the weight or the delay even as a function of the cell properties. For example, if you want to set a delay that depends on the distance between the pre and postsynaptic cell, you can do that easily. You can specify a function, an expression that depends on the distance, or a length that depends, a strength that depends on the cortical depth of a cell, which is some feature that we've also seen, and you can see a diagram here showing that the strength of a connection depended on the depth of the cell. So with NetPyNE, you can define these functional properties very easily. We also have support for learning mechanisms like STDP and RL, and support for gap junctions. In the same line, in terms of connectivity, we also have connectivity at a more lower level, so at the level of the dendrites. So how do you define the distribution of synapses along the dendrites of a single cell? There's, for example, some experimental data that we had available for that, which provides a sort of distribution, a probability distribution, saying, for example, there is a higher probability of synapses being at the soma than at the apical dendrites. So you can provide this data directly to NetPyNE, and the synapses of a single cell will get adapted based on this experimental data. And they will also get adapted to different cell types. So the same probability distribution can be applied to cell types, for example, with different number of compartments.
Oh, it's, I'm sure everybody's finding quite remarkable how you can cover all these different scales using NetPyNE. Let me take the opportunity to remind people that are watching this that you can ask any questions using the GoToWebinar software, and we will answer the questions at the end of the presentation with Salvador. So if you have any questions about any of these different scales and about how you can use NetPyNE to define them, feel free to ask any questions, and then we will cover them. So taking it from here, Salvador, can we now probably what it makes sense is to explain people that are watching how you go from these high-level specification that you define to a specific network instance that then can be visualized and simulated.
Yes, exactly. So once we have defined all these high-level properties of the network that we want to create, we can create specific instances. That means, for example, translating a rule that specifies a probability of connection of 20% between two populations into all the specific connections, single connections from cell to cell. So of course, these instances depend on some random seeds, so you can get many different instances for the same high-level parameters. And once this instance is created, what we get is a sort of hierarchical Python structure that covers all the different scales, from populations to single cells to the sections inside a cell and the molecular level. And you can navigate through this structure very easily. You can see the format here. And you can also access the neuron objects associated with each of these elements that are required to run the simulation. So in the past, what the user had to do is create all these neuron objects by himself. Now NetPyNE takes care of all this and then gives you access to all the neuron objects in case you want to modify any of the properties. Of course, once you have this network instance with all the neuron objects, you can just run a single command to run a simulation. And this is by default set up to run parallel simulation. So you can run it on a single core if you want, on multiple cores on your laptop, or you can run on supercomputers. Just with a single command, or if you're using the GUI, with a simple click. NetPyNE will take care of, again, distributing all these cells efficiently, and then gathering back all the simulation results, the spiking data, voltage traces, LFPs if you selected to record them, and then you would be able to analyze all of this data. So what types of analysis that NetPyNE provide? So we have many different types that we have been adding progressively. So you have analysis at the level of the network connectivity. So for example, standard connectivity matrices. You can show different properties like the connection weight, the number of connections. You have different ways of representing connectivity as a stack bar, for example, or just the individual cells and all the connections as lines. And you, of course, have standard neuroscience plots, such as voltage traces, raster plots, statistics plots. You also have information transfer, like grandeur causality or normalized transfer entropy. And all these are accessible with a single command once the simulation has been executed. We have also recently added the ability to record local field potentials. So you can place any electrodes, recording electrodes, at arbitrary locations of your network in the 3D space. And NetPyNE will record the corresponding voltage traces from all these recorded, the corresponding potentials from all these electrodes. And for example, here we have for a single cell where we placed 9 different electrodes across the cell. And you see the LFP signal from each of them. And the same thing in a network here, where we placed 4 electrodes. And you can then also plot things like the power spectral density of the LFP signal or the spectrograms. Once you have run all the simulation and analysis, one thing that you usually want to do is save all this data. So NetPyNE provides many common formats like Pickle, JSON, and MATLAB, where you can save every aspect of the simulation, from the high-level specifications, the actual network, and the simulation results. And importantly, you have the ability to export and import networks from NeurML. NeurML is a sort of standard language to define models in neuroscience, and it allows you to then convert your network models to other simulators, like, for example, Pine or Brian or Nest. And you can also import existing models from, for example, open source Brain, with many existing models that you could just run directly in NetPyNE. We have also developed, as I was mentioning before, a graphical user interface that allows you to perform all of these different functionalities from an easier and intuitive platform. And the GUI is not only useful for students and beginners who are learning and finding it easier and more intuitive to use, but also for experts. It's usually easier to start prototyping your network and see the results quickly. And once you have prototyped like the initial version of your network, you can actually export that to a Python script, and you can keep developing the network programmatically if you want. And of course, the GUI is also useful to actually explore and visualize existing models. So you can load any of the existing models that are available and run simulations or simply explore results.
That's great. And I'm sure, Salvador, people are wondering if we could see a live demo that I was kind of promising at the beginning of the user interface. But maybe while you get ready, if that's something we can do, I would like to launch a second poll to ask our audience what type of computational modeling they do or they are interested in. So let me do this. So again, everyone, you should have a window that appears on the interface where you can select different options. Okay. Still some votes coming in. All right, 3, 2, 1. I will close the poll and we can look together at the results. So the different categories for the poll were point neurons, detailed single cells, circuits, large-scale networks, and multiscale models. So we have large-scale networks leading with 70%. Then we have detailed single cells, circuits, multiscale models, and 11% point neurons. So that's interesting, and I'm also happy to see that there is a good 40% that is already either doing or interested in getting into more multi-scale type of models. So thanks everyone for participating in the poll. Salvador, can we show a demo of how we could use NetPyNE to build a simple model? So maybe starting from creating a specification, visualizing the model and simulating it.
Sure. So yeah, I'm gonna show like live a very simple example just to illustrate how this works. So here you're seeing the graphical user interface. You see here at the top that you have three sort of large sections. Define your network, where you are gonna specify all these high-level specifications we're talking about. The explore your network, which would be the time where you create this instance, the actual specific network from that high-level specs. And the third panel here, which is simulate and analyze, where you run the simulation and have the ability to analyze and plot different things from the simulation results. So here in the high-level specifications, we have all these sub-panels with the different things you can define: populations, cell rules, all the properties of the cells, synapses, connectivity, stimulation. And I'm just going to show a very simple example. So I'm going to create a new population. Now you see it here. I'm going to name this population, for example, E of excitatory, and I'm going to say it has the cell type pyramidal. I'm going to specify I want 20 cells. And now I'm going to go on to cell rules, and I'm going to give some properties to these cells in this population. So I'm going to create a new cell rule that is going to be applied to all cells of the cell type pyramidal. And I'm going to add some sections to this cell. So I click here to add a new section. I'm going to call this section Soma. And I'm going to add some geometry, some shape to this section. So I'm going to say the diameter is 5 micrometers, or 20 micrometers since the Soma, and the length is 20 micrometers. So I'm going to add another section now. I'm going to call this dent for dendrite. And I'm going to make this much thinner, 5 micrometers diameter, and longer, 150. So now I have a very simple network with one cell type. And if I click in Explore Your Network, it says it's going to instantiate the network. And you see we have generated this very simple sort of ball and stick network model. You can show the cells in different colors just to illustrate the difference.
And this is running on your machine.
Running right on my machine, yeah. So I can just zoom in, visualize the network, and I could plot some figures here. Right now it's going to be very simple because there's nothing. So I'm going to show a couple more examples which are a bit more complex. So I'm just going to show a video of them because it would take a bit longer to simulate here in real time. So here I have the video. And what I'm going to show is an important feature, which is actually importing cells. So you can import existing cells from, for example, Hawk or Python cell templates. And you can then visualize them and use them in your network. So here I'm opening the dialog where you select the name of the cell. I'm selecting a cell which is a Hawk file. It's a pyramidal cell from the motor cortex. And I'm selecting where the mod files are. So the mod files are the mechanisms of the cell. Now you see I can visualize all of the sections of this cell. And I can click on any mechanism and visualize the parameters of this mechanism. So for example, here I have for the dendrite 28, I can see all the different ionic channels that are inside this dendrite, and I can change parameters of these mechanisms. I can now click just in Explore Your Network, I created a very simple cell with a network with 4 cells, and I'm able to visualize this very detailed cell, which shows the real morphology of a cell from mouse primary motor cortex. And I can very easily move around, visualize it, and use it directly in my network simulations for any model that I want. Before we go on to the next model, I forgot to show here in the simple example that we had before that we have real-time interaction with the sort of Python backend that is simulating all of this model. So you can, if I click here, as you see, I'm accessing some of the parameters of this network model. So for the population E, I'm accessing the parameter number of cells, and I see here that is 20. So if I go back to define your network, we will see that this is fully synchronized. So if, for example, I click here just to see that we have 20 cells here. If I want to change this programmatically from this Python terminal, I can just click here. I want 30 cells. I click enter, and you see here it's automatically updated on the GUI, and the number of cells has gone up to 30. So it's fully synchronized, and I can do things either programmatically or directly in the GUI. And if I go now to explore my network, I see that there are 30 cells instead of 20.
Well, so people can, and this has been historically something that has always somehow divided the community. People advocating for a user interface that would make things easier to use, and some other people, no, look, I want really to be using an API for what I do. And what you're showing here is that both options are actually possible to get the best of both worlds depending on what you're doing.
Exactly. So the power, the benefits of both worlds. And again, you can also have this feature of exporting to a script, a Python script that allows you to continue work programmatically. So let me just finish with this other example in the video, where I'm going to just import an existing model. This is a simple 3-layer network model with simplified cells. They have 5 compartments each. I'm here showing, for example, connectivity rules. We have 2 simple rules connecting all excitatory cells to all other cells and inhibitory cells to excitatory cells. You can see here in the panel that I have defined relatively complex connectivity rules. For example, they depend on the postsynaptic cell cortical depth. This Y norm means the normalized cortical depth of the cell. And you have this sort of expression builder built in. Here we can see the network. It has about 100 cells, and we can plot some of the plots showing, for example, the connectivity matrix and the location of cells and the connections. Here we have already run the simulation, so now we have available different plots from the simulation results. So here in the top left, it's showing the voltage traces from one of the cells, the intracellular calcium concentration and extracellular calcium concentration, as well as the potassium current in one of these cells. So this simulation also included this reaction diffusion. So we were simulating the diffusion of calcium inside and outside of the cell and how that affected some of the channels. We also included local field potentials. So these new plots that I've shown here include the raster plot in the lower left and some spiking statistics, and the LFP signal from 3 electrodes on the top right, as well as the sort of spectrogram signal of the local field potential. And this last plot, which is showing the concentration of calcium inside and outside of the cell. So we see here that we are covering all of the different scales from the molecular level with concentrations of molecules up to spiking of cells and the local field potential, which is a more large-scale measure.
Well, thank you so much, Salvador, for showing this. I think everybody finding it pretty fascinating. So one thing that is clear looking at NetPyNE is that you could end up with pretty large scale models using it, because it's so easy to define rules and populations that have thousands of cells. And one thing that probably people are wondering is what type of facilities exist for doing things like parameter optimization. So if you want to explore the parameter space of these large models, for instance, using, you mentioned earlier, supercomputers, is these an available option?
Yes, so on top of having the ability to just run single simulations in parallel, we have also the ability of running what we call batch simulations. So this allows you to, for example, match your simulation to actual biology, so to reproduce the experimental data, which is a preliminary step before doing any actual experiments. And also, once you have actually matched your simulation to data, sorry, to actually explore your models, so to experiment with your models. So for example, you were trying to develop a novel treatment, you could try many combinations of this treatment on your simulation. So to do this, what we have is a separate module that allows you to define very easily the range, the number of parameters that you want to explore, and the values of each parameter. We have now included both like standard grid search, like you explore each of the values that you specify, and also evolutionary algorithms, genetic algorithms that try to search for an optimum solution. And NetPyNE takes care of all the tedious task of submitting all these jobs in different environments. So for example, you could submit jobs in your local laptop using MPI, or you can submit jobs in different supercomputers using, for example, SLARM, which is a common workload manager, or PBS Torque. We have tried this on, for example, the XSEED NSF-founded San Diego supercomputer in Comet, and also in Google Cloud. So just to give you an example of how we've applied this and the scale that we are running these type of batch simulations, we started with our local cluster at the university, which had 500 cores. We've scaled up to these scientific shared clusters like the Comet supercomputer with over 7,000 cores. And more recently, we've also run simulations on Google Cloud, which allowed us to run up to 50,000 cores simultaneously, exploring many parameter combinations in a very fast way. Once we have run the simulations, of course, NetPyNE allows you to visualize the results of these parameter explorations. And this, for example, shows a 4 multidimensional analysis, 4 dimensions with 4 different parameters, and what effect these parameters have on, for example, the firing rate of a specific population of the network. So this allows you to come up with conclusions or match your model to experimental data.
Oh, okay, and so this is great. I wonder if now we can talk a little bit about some models that were developed using NetPyNE. If there is some, for instance, like you were mentioning at the beginning, that has taken full advantage of all these different features that NetPyNE has.
Sure. So there's a number of models being developed right now in several labs across the world, and these range from many different regions like thalamocortex, hippocampus, spinal cord, B1, and different phenomena as well. So looking at different diseases like ischemia or stimulation like TMS and TDCS. There's a full list of all the models in this link here. I believe you will make the slides available later. And I want to focus on a couple of examples which I think are illustrative. So this is the model I've been working on for a couple of years now. It's this very detailed model of M1, primary motor cortex microcircuits in the mouse. So I'm basically trying to reproduce in full scale all the neurons and connections inside a volume, a cylinder with 300 micrometers diameter and the cortical depth, which is about 1,350 micrometers. So this volume of tissue contains about 10,000 neurons and 5 different classes of neurons, 15 populations, and about 30 million synapses. So of course, all the the specific data of the different cell types and populations was all based on experimental data. So we have densities of different cell types and populations across the different layers. We have the proportion of different cell types in each layer. And we have all the morphology and biophysics from electrophysiology of the different cell types. So we built all this information into NetPyNE. And we've also added a lot of information about connectivity, but connectivity about in 3 different scales. So we had, for example, data of the connectivity of long-range inputs, so other parts of the brain, and how they project onto the cells of this motor cortex microcircuit. So the different parts of the brain project to specific layers and cell types. We also had a lot of information about the local microcircuit connectivity, how the different cell types, inhibitory and excitatory, connect as a function of the depth of the cell, for example. And we had a lot of data too on the distribution of the synapses, so how the specific synapses are located across the dendrite of the different cell types. So as you can imagine, all this information, it was hard to make sense of all of it, just looking at experimental data. So building a model in NetPyNE facilitated integrating all this and having a theoretical framework to actually experiment with this microcircuit. We got some results from the simulations, which we're now preparing for publication. So here are some, this raster plot showing the activity of the neurons in all of the layers, voltage traces, examples from one of the cells. And we see also, we recorded LFPs, and we found some interesting phenomena like these emerging LFP oscillations, oscillations in the theta and the gamma band, which emerged naturally from the network model. So we didn't build these oscillations into the network model, but they emerged spontaneously based, we assume, on the connectivity data that we used from experimental data and on the biophysical properties of the neurons. Another example, which I think is interesting, is this thalamocortical network, which is a popular model from Podgens and Disman. And it's interesting because mainly because of the scale of it, and it hadn't been simulated before in NEURON. So the network has 80,000 neurons. They are very simple model, simple point model neurons. And it also has 300 million synapses. So this reproduces 4 layers of thalamocortex. And we converted this model from NEST into NetPyNE, and we have been able to simulate it in Google Cloud. And as you can see, we reproduced the original NEST paper results with the same statistics. And the advantage of having it now in NetPyNE and NEURON is that you can, for example, try to modify the neuron implementation to, instead of having a point model, have a more detailed biophysical model and see what effect that has on the network. Of course, you can also just use a lot of the built-in analysis function from NetPyNE to explore this model more in depth. And a final example I wanted to mention is this collaboration with Stephanie Jones from Brown University, which has an NIH grant to build what is called the Human Neurocortical Neurosolver, which is basically a tool to try to reproduce EEG and MEG signals from the brain and try to understand it from a biophysical circuit model. So they take experimental data from EEG labs, and they try to reproduce those signals using a biophysical network model and try to interpret then the different signatures of these signals and how they associated to disease and normal function. So they contacted us, and we've been working together to convert their biophysical network model, which is a thalamocortex network, into NetPyNE. And the objective of this is to make it easier in the future to, for example, modify this network or increase the scale of the network. And NetPyNE, of course, provides a very easy framework to do this.
Well, this is quite interesting thinking. We might want to organize a webinar specifically on these. But this was a great showcase of all the different models, Salvador. I'm thinking, so there's been a number of questions so far that were asked. Before getting to questions, I would like to launch our third poll to essentially ask you, of all these different features that you've seen today, what are the ones that you find most interesting or useful for the type of work that you do? So you should have, once again, a window appearing on your screen with different options. And give it another few seconds. All right. Thanks everyone for voting. Let's have a look at the results together. So 73% for the graphical user interface. Then we have network specification, 47%, the network generation instantiation, and then we have 1/3 with the parameter search via parallel simulations. Thanks everyone for voting. And before we go to questions, I want to take a first opportunity, Salvador, to thank you so much for the presentation thus far. I think it's been super informative for everyone. And maybe before we go to questions, one last thing, if we can share some links to point people at where to start if they want to use any of these that you have shown today.
Sure. So we have like very comprehensive documentation online. This is the web, netpyne.org. And you have a tutorial, a step-by-step tutorial that you can follow with all the examples, and also how to use the GUI. We also have 2 question and answer forums, Q&A forums, that already have lots of questions, so you can ask there if you have any issues or search the existing ones. And here is a general list of useful links, the lab website, the NetPyNE website again, the NetPyNE UI website where you can install it, and our GitHub repo, which of course all this project is open source, so we welcome any contributions from people or suggestions. And here's a list of our funding agencies and the different institutions that collaborated in this project. Thanks, Mateo, for organizing this.
Wonderful. No, thank you, Salvador, for what has been a fantastic presentation. So I would say let's go and try to answer some questions. And in the meanwhile, if people want to ask more questions, we have 7 minutes before the end of the webinar. So let's start with the first one. Do NetPyNE or its dependencies work with Python 2 and or 3 or both? So what is the current state of the compatibility with the different versions of Python?
So we have support for both Python 2 and Python 3. We have recently switched from making the development by default from Python 2 and Python 3, but we still support both. And the website describes how to install the version that works for either of both of those versions.
All right. And if there is any conflicting information anywhere, it's probably because this switch has been very recent, was just last week, right, Salvador? So, so yes, that's the current state of things. Another question, I think it referred to when you were showing tutorials 2 and 3 about how was the network activated in those examples, or if it was spontaneously.
So activated, I assume, means instantiated and simulated.
I was thinking in terms of like inputs to the network.
Okay. So yeah, so both of those networks have what we call external inputs or background inputs, which is usually what we do in modeling to simulate activity arriving from other parts of the brain, which sort of gets the network going and provides some background inputs to start firing. And of course, there's the local connectivity, which allows it to fire in different patterns as well.
Okay, and there's more and more questions. One, in any of the, does any of the data for the simulations, so I'm assuming for any, for instance, of the models that we've seen have been built with NetPyNE, does any of the data come from a three-dimensional volume scanning microscopy that you know of?
I believe some of our data from the M1 microcircuit network, some of the morphologies come from microscopy, two-photon microscopy, and I'm not sure about all the other projects that are being developed. But yeah, you can check the list and maybe contact some of the authors if you're interested.
Sure. And another question: are you aware of any public data sets that might be in public databases that can be used with NetPyNE?
I mean, in general, any public dataset can be used because you are converting any experimental data onto these high-level specifications. So there are many, like from CRCNS, and of course there are many existing models available. For example, in ModelDB, you can find existing cell models of many different cell types, and those can be directly imported into NetPyNE. But in general, any dataset that you find, you can easily convert it into this high-level specification format and create connectivity rules that match the data that you're seeing.
Okay, and one last question. In terms of if you think GliaCell will be involved in any of those models in the future?
So I believe the reaction diffusion module in NEURON is actually developing some features to reproduce the processes that involve glia cells. So those will be readily also available via NetPyNE.
All right. So we are pretty much at the end. So I want to thank you again, Salvador, for this fantastic introduction to NetPyNE and for the demo. I want to thank everyone that has been listening to this. I hope you found it interesting. So Salvador will be presenting NetPyNE at the Google Next conference in London in a few weeks' time. I know the registration is not open for everyone, but there is some invites that Google sends to people. So if you're lucky enough to have one of those, then you will be able to also listen and learn more about NetPyNE. This is pretty much everything from it. Again, thank you so much, Salvador.
Thank you so much, Mateo, and thanks everyone who came to listen to the talk. If you have any questions, please contact us directly, and thanks again.
Sure. And don't forget, we have a Twitter account if you want to stay updated with next activities, next webinar. It's at MetaCell, so you can follow us to just be updated. Thanks again for your attention, and we'll talk to you soon. Have a great rest of the day.